Well, look, welcome everyone. So many familiar faces and names today, and welcome to anybody who's new to our symposia. I'm Jeffrey Guy, the founder of the Guy Foundation. We have Nina here, who is basically in charge of looking after all our arrangements and is our program director. And I'm going to hand over in a little while to Alistair, our director of science. This is the eighth of our online series that we've done since our inauguration about 5 years ago, and this series is dedicated to quantum biology and space health. Now, 2 weeks ago, we had our first symposium in this program, in this series, and our speakers were Tom Marshburn, an astronaut, who gave a fascinating talk and account of sort of the day in the life of an astronaut. And that was followed by Scott Smith from NASA's Johnson Space Center Nutritional Biochemistry Lab for Space Health. Today, we're going to be talking about mitochondria. And I think for many of you, you will know that we believe mitochondria sits at the heart of the central bioenergetics and the interaction of bioenergetics and quantum physics. And that's the basic fundamental of some of our thinking in terms of quantum biology. And we think trying to understand what's going on with mitochondria, both on Earth and in space, will give us some great insights into, one, the pathologies we might be encountering in due course, but also therapeutics and therapies moving forwards. Clearly, space provides an environment of vastly different physical attributes in terms of the electromagnetic and gravitational fields, amongst other things, of course. So I really want to extend a really warm welcome to Doug Wallace and Afshin Beheshti. Alistair is going to give a more precise welcome, but I'm really excited about today's presentations. I've had a glimpse at the slides. They look fascinating. We're going to depart again from our normal structure where we normally ask both speakers to speak and then have a roundtable discussion over both of the papers presented or the presentations presented. Doug has another commitment in New York in about an hour and a half. So we're going to ask Doug to present his talk, and then we'll have Q&A on Doug's presentation, and then we'll move into Ashwin's talk. So as you know, these talks are recorded. We prefer you keep your questions to the Q&A session. We record the Q&A session purely for our own, making our own notes. They are not broadcast on YouTube or on our website. If somebody completely and totally loses the plot and you want to ask a question as going through, I'm sure the speakers wouldn't mind being interrupted, but I prefer if you did keep your questions to the end. Without further ado, I'm going to hand over to our Director of Science, Alistair. Alistair, over to you if you'd like to put your slides up. Thanks.
Thanks, Jeffrey. My name's Alistair Nunn. As Jeffrey said, I'm the Director of Science for the Foundation. I also hold a visiting chair at the University of Westminster in quantum bioenergetics. Well, it's a title anyway. Okay, so why mitochondria? So I'm a slight departure here. I always think about these things often from the science fiction point of view. And just as one of the interesting areas that science fiction often predicts a lot of these things. And certainly one of the big franchises I think we're all aware of is Star Wars. And George Lucas talks about these things called midichlorians, which actually, if you investigate them, are actually mitochondria. But these little entities in Jedi, which enable them to detect the force, the life force. And they certainly are able to detect and perhaps created by energy fields. But of course, it goes further than that. It also in science fiction, Doc Brown developed this idea of the flux capacitor. Now, we all know this as a sort of, temporal shifting device, but actually Nick Lane has also used this to describe a mitochondrion. And I think, Doug, actually, you mentioned the capacitor concept a bit later on. So one of our approaches has always been to start right at the beginning. And this means, the beginning of life. Life obviously started on this planet from what was available at the time. So as the Earth cooled, there was a bunch of fundamental chemicals about, and of course there were the laws of the universe in relation to physics and chemistry. One of the key aspects of this seems to be charge transfer. And certainly one of the strongest theories about the origins of life, and I know there are other theories out there, as the RNA world, there's other theories, but certainly this is one of them, is that it may have started in an alkaline thermal vent. And the importance of this is this provides a very large energy potential where upwelling hydrogen meets seawater full of carbon dioxide. And one of the key potential molecules involved in this is iron sulfide, which has some interesting quantum properties. Anyway, this seems to allow the formation of what was effectively a dissipating, self-organizing, far from equilibrium structure. And if we move forward a few million years, we come to start to understand the idea of the electron transport chain. And obviously, this is where Peter Mitchell starts talking about the chemiosmotic theory. And this is all linked into the Krebs cycle. And certainly the earliest life forms seem to be prokaryotic. And not very long afterwards, photosynthesis also evolved, which led to more oxygen. And then, of course, we get into free radicals and everything. But certainly all of this can also be described from at a quantum level. But for today's talk, we're more and more interested in the mitochondria. Now, one of the key events in evolution certainly seems to have been that an archaean and a bacterium got together and to some kind of endosymbiotic event to form the modern eukaryotic cell, which enabled complex multicellular life to evolve. Cool. Sorry, that's my phone going off. And certainly, which means that the mitochondria is actually central to a lot of what's going on in life today. So we can often use the origins approach to understand how disease occurs in today's world, and certainly also leads to understanding of aging. But it also raises some very interesting concepts because, as most of you have been attending these lectures will know of the work of Mike Levine and the morphogenetic field. But of course, the morphogenetic field must have come from somewhere. And certainly when we look at charge transfer, we can apply physics straight away. And of course, charge transfer means you generate fields, and these fields will interact with the charges themselves. You can start to see how a field matter organization could have been important at the beginning of life. And this could have led certainly to the formation of basic ion channels and other protein structures. And so through evolution, we get to more and more complex entities. And I'll just put in here this little picture here. And this is actually thought to be a relative of perhaps the ancient Archaean, which life started from. And the interesting thing about this prokaryote is it's not a modern cell. It has a very primitive cytoskeleton, and it can also modulate its membranes. And this begins to help to explain why these two bacteria and Archaean got together. But we're obviously interested in the space research. One of the ways, perhaps, of looking at this, of course, is that we evolved and life evolved in a set of circumstances where certainly gravity, magnetic field, okay, changing direction potentially, and certainly circadian rhythms were there all the time. Obviously, pressure and oxygen probably changed, and oxygen increased through evolution, but radiation had an effect. And clearly, this canalized life into like a Goldilocks zone of where it functions. And within this, it can adapt. And we know this is the principle of hormesis, where what doesn't kill you makes you stronger, and it probably makes you smarter as well. And this is sort of expressed through the electron transport chain, membrane potential, and the production of ROS, which of course is most strongly seen in the mitochondria. But of course, when we go into space, all of this swaps around. And of course, in our tin cans, we can control for pressure and oxygen, but we can't really deal with circadian rhythms. We certainly can't control gravity at the moment. And well, there's obviously an interaction with magnetic fields and obviously radiation. And we're not canalized over here. So we're quite outside what we would call our flight envelope. And I'm sorry, to the many people out there who don't understand the flight envelopes, but engineers will. I'm a pilot, and one of the things we have to abide by is the design limitations of the craft we fly. And most of you will know if you fly an airliner too slowly, it'll stall. And if you fly it too fast and pull very hard and stick, you break the wings off. So we have an operating zone. And this seems to be pretty much how life has it, as a metabolic operating zone. And at Westminster, we've always been very, very interested in what is optimal health. We often talk about disease, but what actually does health mean? And we start to look at statistics like, well, how long can humans live? And the evidence is, not much beyond 120. And there's some new thermodynamic theories that suggest maybe not much beyond 130. But when you start to look at what we mean by a healthy life, we see might well be that most humans, I mean, obviously give and take genetics and epigenetics to some degree, could live to be near 100. And what's key in this, most humans that live this long often live in good health for a very long time. And everything goes wrong at the end, and then they suffer, they fall off the mortal coil. And this is kind of called morbidity compression. But actually, what we see in most societies today is, of course, we don't live that long. Life expectancy is nearer 80, 85. We start getting ill around 77, 75, 70. And in some populations, in particular those that are suffering a modern sedentary lifestyle, they can often not live much beyond 75, and they have a very greatly extended period of ill health, which is called morbidity expansion. And you go even further into stress, and of course we don't survive. Now, I'm not going to go into this, but of course this could be seen as an adaptive response to churning over the population. But of course, when we go into space, are we starting to see this, what appears to be an accelerated aging syndrome? And this is something which has been predicted for quite a long time. Now, one of the most obviously researched areas is what we look at when we talk about reduced gravity. And mitochondria are often seen or depicted in the press and everything else as being these little organelles like batteries which produce energy and don't do much else. But actually, they're highly integrated to the entire function of the cell, as you would predict from the origins theories. And this means that they're integrated with the cytoskeleton. So any change in the shape of the cell is going to feed back into the mitochondria and vice versa. And this is seen in many different ways, certainly in many conditions. That's related to inflammation. We certainly see it in things like neurogenic problems and certainly in insulin resistance. And one of the key, very well described biochemical interactions is to do with tubulin and the cytoskeleton. Of course, tubulin, as many of you know, is key in the cytoskeleton. And it exists as this big polymer, but it can also exist as a monomer. And this can obviously interact with things. One of the entities proposed to interact with something called VDAC, which is absolutely essential for mitochondrial function. So there's clearly a relationship here to cell shape and gravity and stress. But of course, this isn't just about the gravity. We also know that electric fields have a lot of effects. Certainly they affect the modulate, and they can affect the mitochondrial cytoskeleton interaction. And certainly, in terms of theory, it's predicted there may be effects, for instance, from Froelich condensates involving the cytoskeleton and the mitochondrial electric fields. And more recently, Philip Curian and his colleague Celerado have been looking at superradiance. So there's all sorts of very interesting quantum things going on here. But this isn't the only thing going on. There's another thing which I think a lot of people don't quite realize is the role of membranes. And of course, membranes are probably very ancient, are key in life in terms of being able to separate charge. But they are, in fact, or can be viewed as very thin capacitors because they have some degree of polarization. And as soon as you change the charge across the membrane, you alter its shape very slightly. And this is called electromechanical coupling or electrorestriction. And there is also thus a close relationship to fluidity. And now, as any biochemist will tell you, fluidity in a membrane is absolutely essential function. If it comes too crystalline, i.e. it gets too stiff, it doesn't work. If it goes too fluid, it falls to pieces and it doesn't work. So there's a very narrow, like a Goldilocks zone where it works. And of course, this is also controlled by redox. If you oxidize membranes, they get stiffer. And so nature has spent a long time trying to get this right. And of course, as I think Doug will obviously understand this and be able to explain it much better than I, the mitochondrial membrane has some very interesting fluidity properties, and certainly around cardiolipin. But this raises some quite interesting concepts about the origins theories and the evolution. We can certainly say that this is a very simple diagram where this is a cell which has been slightly squished by gravity. And we know that hypergravity, so increasing G, could actually cause adaptive responses, strengthen the whole system. And indeed, through our evolution, running around, jumping up and down, lifting weights and things can stress the system and compress it a little bit. But what it probably never did was be exposed to continual reduced G. It's just that cells never been exposed to that. There may be slight differences in animals and creatures in the sea because of buoyancy. But for most of us land-based animals, falling off a cliff was about the only experience we'd ever have of reduced G. And of course, it tended to terminate very quickly, and we never evolved beyond that, unless you evolved wings, of course. So this area of adaptability seems to be, we don't understand what happens in reduced gravity. And certainly from the foundation, as Jeffrey mentioned, we're very interested in the mitochondria, and we view mitochondria as the canary in the coal mine, or the metabolic coal mine. And all these factors can interact, and what happens to mitochondria is reflective of a system. And so what the mitochondria does also is reflected in how the system adapts. And of course, we can start to look at this from the quantum perspective, and especially when we start thinking about fields and potentially the role of circadian rhythms and certainly gravity. So in summary, an origins perspective suggests that both gravity and electric fields are key in how life works. And this process will have been canalized over billions of years. And in fact, we've kind of been slave to it. And of course, the mitochondrion could be seen as this kind of canary, which is just telling us what's going on. And certainly, as both Doug and Afshin will be saying, multiple studies have shown astronauts display mitochondrial dysfunction. And this could be associated with an accelerated aging syndrome. And of relevance to medicine, of course, is that, and certainly for space research, it's now becoming very clear that the to go into space boldly, we probably need to have very good health. The problem is, of course, we don't fully understand it on Earth either. We don't fully understand what ill health and aging is yet. So we're kind of going to space with one of our hands tied behind our back. We may be able to build very nice spaceships, but do we fully understand the physiology of the astronauts in them? And one of the interesting things here, and I just throw in another kind of angle of this, is of course it maybe nobody else has either. And of course this relates to the astrobiology idea. And we published this paper many years ago as a kind of a parable. And this is the fact that if you try to explain why it is that modern lifestyles, sedentary lifestyles, seem to accelerate the aging process, you could argue that it's the removal of the very stress factors that led us to evolve in the first place. Of course, when you develop technology, you think, right, I want to live in a very comfortable lifestyle. So what do? Well, you remove the need to move around. You live in a nice warm house. You have as much food as you like. And indeed, you also change the food you eat. And so we suggested that one of the explanations of Fermi's paradox, and I think most of you were familiar with Fermi's paradox, but this is something that Enrico Fermi back in the 1940s and 50s was supposed to have said. And this was an age when everybody was looking out into the universe, realizing there were billions of galaxies and stars, thinking, well, it must be aliens everywhere. Well, where are they? And he's basically said, well, where are they? And because we haven't seen them yet. And so this led to all sorts of theories, including that nuclear annihilation, space travel was impossible. And we've now thrown our lot in here and said, well, actually, what happens is the aliens can't afford their healthcare costs, so they can't afford space travel. There are some implications, certainly from this, is that just how good are the models on Earth for mimicking zero gravity? I suspect they're not very good. Magnetism we might be able to look at, but I certainly think the best model of space travel is going into space. And also the type of organism we study may be very interesting. It's something that can evolve very quickly, like a bacterium, very short life, probably will adapt very quickly, but it's a very complex organism which relies a lot of cooperating cells stacked on each other and are also relying on the gravity to keep that all together, may suffer more. Anyway, without further ado, I'll pass you over and introduce first Professor Doug Wallace from the Chop Research Institute. No real introductions here. He's one of the world's experts in quantum mitochondria, and he's going to be talking about metabolism in space, and certainly some very interesting data on how there may be electromagnetic coupling between mitochondria. And this will be followed by Dr. Afshin Beheshti, a PI at Blue Marble Space Institute of Science. And he's again going to talk about mitochondria but what happens in space, and in particular, what happens with RNA. And he's going to talk a little bit about birth defects and countermeasures. So over to Doug.
I really want to thank Jeffrey and Alistair for, one, the invitation to be here, for the wonderful symposiums they've had, and Alistair for your deep understanding of biology and the origin of life and its relationship to mitochondria. So I've been very pleased to be able to participate in some of these symposia, but I also have the privilege and honor of being a colleague and I hope a friend of Afshin. So we've had many fun conversations. So I was going to take advantage of the fact that the discussion here is about quantum biology and space flight, and I put in the center of that connection the mitochondria, as has been so beautifully mentioned. So why did 50 years ago I decide to study the bioenergetics of human health? Well, coming from a physics background, it seemed very logical that life couldn't just be about anatomy, which I was completely told that was all about medicine, but also whatever made anatomy animate, which has been known for a couple of thousand years as the vital force, but that's really energy. And you need information both about energy and information about anatomy. So it seemed to me that studying the genetics of energy might be very important in understanding life and the health process. And how did we look at the genetics of energy? Well, we need to look for where the energy is coming from. And the energy is coming from the mitochondria, which basically burns hydrogen from our food with oxygen in our environment to create a capacitor, as Allison so nicely indicated. But what's important to think from an evolutionary point of view is that in general, energy is limiting. All animals will maximize their energy so that they can then optimize their evolutionary success. So How does this all then work in the quantum field? Well, mitochondria generates energy by a process called oxidative phosphorylation. And as I hope to show you, oxidative phosphorylation is all about the flux of electrons and protons in this capacitor system. And what's interesting about both electrons and protons is they have a nuclear spin of 1/2. So spin then is very important because spin is influenced by magnetic fields. So therefore, evolution had to be thinking or had to be acting within the context of quantum biology because it's acting on magnetic fields of the Earth in relation to spin systems of both electrons and protons. So what is this magnetic field that we've all of this system evolved into? And as you just heard, it's about 50 microtorr, which is the magnetic field. The concern today is that if we get far outside of the Earth's magnetic field, there won't be any magnetic field. So the question is, will oxidative phosphorylation still be optimized when there's no longer the context of 50 microtorr? And so that's basically what I hope to discuss with you. So let's go back to what Alistair said. Mitochondria is really a symbiont. It arose in the order of 2.5 billion years. It had its own genome, which was co-equal with the archaeobacteria that he discussed. But many of the genes, in the order of 1 to 2,000 over evolutionary time, were transferred to the mitochondrial DNA into the nuclear DNA, where they're distributed across the chromosomes. What the mitochondria retained is a circular molecule that codes basically for the wiring diagram of the mitochondrial power plant. And that mitochondrial wiring diagram involves 13 polypeptides, 7 of which go to the first enzyme of energy production and oxidative phosphorylation called complex 1, of which there are 45 proteins. So 7 of the 45 proteins from mitochondrial DNA, 1 of the 11 proteins for the next enzyme, complex 3, 3 of the 13 proteins from complex 4, and 2 of 15 proteins of complex 5. And the mitochondria has its own protein synthesis system to convert the RNAs into peptides. So the mitochondrial system then uses these polypeptides to generate these enzymes, complex 1, coenzyme Q, complex 3, cytochrome C, complex 4, and then complex 5. And the way this worked is hydrogens, let's say coming from glycolysis, would generate pyruvate, which could be reduced to lactate or get an amino group to give you alanine, which are basically characteristics we see in patients with severe mitochondrial dysfunction. The pyruvate goes through the pyruvate dehydrogenase to the TCA cycle, which strips the hydrogens off the hydrocarbons, putting them on the carrier NADH, and then the hydrogens are burned by complex 1, Q, complex 3, cytochrome C, complex 4, to oxygen to give water. And as the electrons are moved down this electron transport chain, they put protons out across the membrane to give you an electrochemical gradient, which, as Alistair mentioned, was originally hypothesized by Peter Mitchell. And that membrane potential is then used by the ATP synthase to condense ADP and phosphate to make ATP. And that is exchanged across the inner membrane by the adenine nucleotide translocator, where it's then used to do work. Anyway, the point is that then we're burning hydrogen with oxygen. We're generating a capacitor. The capacitor is used by the ATP synthase to convert that into chemical energy, which is then used to do work. And since it's basically a furnace, there's always smoke, and the smoke are single electrons that are transferred to O2, give you first superoxide anion, which is converted to hydrogen peroxide by manganese SOD, and hydrogen peroxide is converted to hydroxyl radical if it gets another electron, and these are called the reactive oxygen species. They can be detoxified by taking hydrogen peroxide to water, but this is rate-limiting step. So basically, we build up hydrogen peroxide in our mitochondria. Now, if the mitochondrial energetics become damaged, the membrane potential declines, more electrons are transferred to O2 to give more reactive oxygen species. This plus calcium uptake in the mitochondria impinges on the permeability transition pore. that opens a channel which causes the short circuit of the membrane potential. The intermembrane swells. These stored apoptotic proteins are released through Baxen-Bach channel, and they then degrade the cell by apoptosis. So basically, the mitochondria then is involved in generating energy. It regulates redox balance. It generates reactive oxygen species, regulates calcium. But important for this discussion, when these things are out of balance, it results in cell death. Now, the mitochondrial then genome involves then the 13 polypeptides in the mitochondrial DNA, somewhere in the order of over 1,000 nuclear encoded genes, of which about 150 to 160 are involved in assembling and regulating oxidative phosphorylation. And this is just the mitochondrial nucleoid inside a mitochondria by our picture with a ARIScan microscope. Okay, so let's see. All right, so make sure that I'm on the right slide, right? So basically then, how does this system relate to what we're going to discuss today? And so here, basically, we have oxidative phosphorylation, and generally that's important in post-mitotic tissues because it generates a lot of ATP. And the brain is 2% of your body weight, uses 20% of all your mitochondrial energy. So any subtle changes in oxidative phosphorylation are going to preferentially affect the brain, followed by the heart, the kidney, the muscle, endocrine system, et cetera. Now, if you impair mitochondria, as I said, you increase reactive oxygen species, superoxide anion, hydrogen peroxide, and hydroxyl radical. If there's within normal limits, that will modulate the enzyme HIF1 alpha, and HIF1 alpha has a transcriptional program that then generates the genes for glycolysis. So you have then a feedback system to OXPHOS that balances this system. And if you are not in a post-mitotic system, but you want to start making things, then you have to go into anabolism, and that then is done by modulating OXPHOS to alter reactive oxygen species to drive glycolysis to generate those phosphate intermediates to assemble cells. However, if the OXPHOS is too damaged, you get high levels of these systems, and they then activate the permeability transition pore and damage the mitochondria that then releases into the cytoplasm and into the bloodstream what are called mitochondrial damage associated molecular patterns, one of which is the mitochondrial DNA. The mitochondrial DNA then interacts with the inflammasome, C-Gas-Sting, and the toll-like receptors to drive inflammation, and when that gets too severe, you get then cell death. So in a healthy individual, we have high OXPHOS, low reactive oxygen species, low stabilized permeability transition pore, low mitochondrial DAMPs, and no cell death. But in impaired OXPHOS, you have then decreased OXPHOS, increased ROS, increased permeability transition, increased DAMPs, and increased damage. And if you look at some of the early studies that were done on humans, twin studies that I'm sure Afshin will talk about, one of the first things that was observed was that the oxidized mitochondrial DNA was very high post space travel, but was not so oxidized in the pre-astronaut phase. And what they then saw is in twin studies that the mitochondrial DNA transcripts in post space flight had marked increases in the mitochondrial DNA transcription. So why would they be doing that? Because the nuclear encoded OXPHOS genes, as you'll see, were badly inhibited, and the mitochondrial DNA was trying to compensate by upregulating the mitochondrial DNA transcription, which I hope to convince you was the case. So let's see. So this is some studies from the human analysis. And what we're looking at here are peripheral mononuclear blood cells. And these are different types of blood cells that are involved in the immune system. And the important system is if you look at the transcriptional profile from these cells, what you always see is oxidative phosphorylation is downregulated in every one of the cells. And if you get more data, and depending on the number of cells you can get, you will see that that involves all of the OXPHOS complexes, complex 5, 4, 1, et cetera. So there's a coordinate downregulation of oxidative phosphorylation, primarily occurring through nuclear gene transcriptional regulation. And if you then just look at the aggregate peripheral blood cells, you'll see that complex 1, 2, 3, 4, and 5 are all downregulated. You see this upregulated mitochondrial biogenesis, which is the induction of mitochondrial DNA transcription. Mitochondrial ribosomes and oxphos are downregulated. And what's particularly interesting is you see then the upregulation of both inflammation and mitochondrial damps, and that then results in, activation of apoptosis, panoptosis, necrosis, that is the progressive loss of cells. So together with Afshin and our colleagues, we've been doing bioinformatics analysis, of rodent, and particular mouse, RNA transcriptional data. And so this is the classic C57 black 6 mouse. And here we're going to, again, first look at the immune systems, the spleen and the thymus. And what you immediately see after a prolonged space exposure by these mice, you see downregulation of all the oxphos genes. You do now see this upregulation of mitochondrial biogenesis, which is, again, the mitochondrial DNA, and also a nuclear encoded set of genes called coenzyme Q. You see exactly the same thing for the thymus, where, again, you see the mitochondrial DNA upregulation as being the compensatory response. And then if you look at the HIF1 alpha, you see that that's increased in the spleen. You see glycolysis goes up because that's what you'd expect. You see then activation of the integrated stress response and of the damaged mitochondrial damps. But in the spleen, interestingly enough, they inhibit apoptosis, whereas in the thymus, we see just the increase in HIF1 alpha, the massive increase in cell death. So we're losing T cells, whereas the spleen is trying to retain the B cells. If we then look at what happens to these mice in spaceflight, if we look at differentiated tissues, if we look at the eye in bulb C mice, we again see the massive downregulation of mitochondrial function and the induction of mitochondrial DNA. We see that same thing in a postmitotic tissue, the kidney, and again with the mitochondrial DNA being upregulated. We see then in the kidney the upregulation of HIF1 alpha and glycolysis. We see the upregulation of the immune system and all the apoptotic systems that are killing the postmitotic cells. Interestingly, in the eye, which is immune protected, there is a retention of some of the immune function and only partial upregulation of inflammation and cell death. So how does all this relate to microgravity and oxidative phosphorylation? And so to discuss that, we need to go back to what is happening in the mitochondria. So we're burning hydrogen by NADH to NAD through complex 1, which, by the way, as the first initiation is the electrons are moved to a flavoprotein, FMN. Electrons move through complex 1 to CoQ. They go to complex 3 to complex 4, and these are pumping protons from complex 1, complex 3, and complex 4 into the across the mitochondrial membrane. These protons, however, do not just stay there. They can either go through the ATP synthase, but often in a sedentary individual, all the ATP is already phosphorylated. So what happens to all this proton buildup? What we discovered, and by making a mouse that's deficient in the adenine nucleotide translocator, which exchanges ATP and ADP, was the original idea. What we found out is that it's a lipid-gated proton channel. So when the membrane potential gets very, very high, then the ANT opens a proton channel and completes a circuit. So if we then look at a patch-clamped mitochondrial intermembrane, and we change the membrane potential, that is the potential across the membrane, we can see that when we have a normal mitochondria, we increase the flux of protons. If we knock out the adenine nucleotide translocator, very annoying, we completely block that effect. So then that says that the adenine nucleotide translocator is the rectifier of the membrane transport system. Okay, so how does that become interesting from the point of, oh, by the way, and I just want to mention again that both the electrons that are moving through the system have a spin of 1/2, and the protons that are being pumped also have a spin of 1/2. So this system is uniquely structured to interact with magnetic fields. That's a key point that I wanted to make. So somehow I have to get this to change. Okay, so now what would that mean? Well, what we wanted to know is where the protons are located. So these are cristae that are in foldings of the mitochondrial inner membrane. So they go in like this, and then they come back out. So that tiny little volume in each of these structures is somehow where all the protons are pumped, because all the electron transport chain are on the membranes of these cristae. So they're pumping protons into this tiny little space. So we wanted to find out if that was in fact true, because that seemed most astonishing. So basically, the idea of the model would be that you have electron transport chain, and it's taking protons from the matrix and pumping them into the cristae, which have sealed by OPA1 and the Micos complex. So all of the protons are locked up here. So to test if that was true, first thing you can do is put in what's called an uncoupler. So we glued mitochondria, and this is my colleague Peter Burke. We glued mitochondria with an antibody to a graphene film. The graphene film, if you put a membrane electrical potential across it, a graphene film becomes a wonderful pH indicator. So if we then just open all of the membranes to movement of ions, first thing that happens is the membrane potential collapses. That's not surprising. All of the hydroxyl ions from the matrix come out into the space around the mitochondria, and the pH goes up. Fine. But then what we did is we used an enzyme called BIM, which is an apoptotic protein that then cleaves OPA1 and would open then this block in the cristae lumen. And then what happened was most remarkable and had never been observed before. Immediately on adding BIM, the pH didn't go up, the pH dropped like a stone. So what that means is that even though the matrix retained the hydroxyl ions, mitochondrial cristae released all the protons. So that proves that all the protons are in here in some kind of physical state that has almost no water. So what kind of physical state could that be? That's a major area of our interest in research. See if I can change my... So if we now look at the mitochondria in adjacent forms in a heart, what we see is that all the cristae are aligned with each other, even across mitochondrial outer membranes. So that suggests to me, as we just heard from, I think it's Reese Mole, that there must be some kind of interaction between adjacent mitochondria that can't be due to ion transfer, because the outer membrane is not allowing those ions to move. So what would that do? That would cause then a flux of protons from the electron transport chain into that cristae lumen, whatever it is. And then when the membrane potential gets too high, opening through fatty acid intervention, the adenine nucleotide translocator, creating an oscillated circle. So here we've created mitochondria, and we've used a dye that is monitoring the membrane potential. And we've put the mitochondria down this small tube, which is 5 micron by 1 micron. We're monitoring visually the mitochondria. And you can see not only when we add substrate, the mitochondria come on, but then they oscillate. So we wanted to know if that oscillation was in fact real electrical. So we created a structure where we have electrodes, and this is the thin tube on which the mitochondria are moving through. So this would be the tube, these are the electrodes, and we've added carbon nanotubes as sensors for membrane potential. So as the mitochondria comes along, it crosses the the field, and of course the field is disturbed. But then what you see is this oscillation. Then the mitochondria goes back, another one comes in, and oscillation and back. And if you look at this in the fine structure, you see this area of rapid oscillation of the membrane potential. And so that must be occurring. We believe that's occurring to this fluctuation of the proton field. So two things are then going on. One, electrons are moving through the electron transport chain, and this is not our work, I think is very germane, from the Osman paper. I think we have one of the people here in this group. And they then conjectured that electrons could go into the flavine mononucleotide in complex 1, nicotinamide mononucleotide in the complex 1 and nicotinamide adenine dinucleotide in complex 2. The idea would be that an electron enters this multi-ring structure as a single electron ready to go on to the iron sulfur centers of complex 1, but it also has the capacity that the second electron might move in. And so that creates the possibility of having then a singlet, triplet electron mixing that. And so what they did is they put in either parallel or perpendicular oscillations equivalent to oscillation of electrons in a magnetic field. What they found is that changed the production of superoxide anion and hydrogen peroxide. And what's really remarkable is if the oscillating field, the electron oscillation, was perpendicular, then it didn't affect mitochondrial respiration, that's OCAR. It did affect mitochondrial lactic acid production, which is glycolysis. Whereas if they had parallel fields, it affected oxidative phosphorylation, but did not affect glycolysis. So what that says is this electron flux could well be affecting mitochondrial energetics. And I'd like to suppose, although we don't have data, that it could also be affecting the proton flux. So here we have then another study from Marino and Costello, and they then shielded cells from a magnetic field. And what they found is that it changed mitochondrial ROS production. So that, again, is either to the flavines or perhaps protons. So our model is that the mitochondria is using electron transport chain to put protons in. The protons are used in the Christie lumen to drive ATP synthesis. But this is almost no water, so we have a feedback system of oscillation. Would that create magnetic fields? So this is a study that was done by my friend Tim Roberts at CHOP. And he works with this machine, which is called a magnetoencephalograph. If you put a child in this field, you can measure the magnetic field oscillations coming from that child. So this is an individual with normal magnetic, a normal individual that's being exposed to auditory signal. And we see this big oscillation of the magnetic field. In an autism patient, we see that that is lost. Okay. So why is that important? Because we've shown that mitochondrial DNA variation, which we call haplogroups, is a major factor in risk assessment for autism. And so one of the things that we wanted to do is take advantage of the fact that there are mitochondrial DNA mutations that affect oxidative phosphorylation. So this mutation in the mitochondrial DNA ND6 gene, which affects proline 25 to leucine, causes a neurodegenerative disease. So we created a mouse in which we imported that same mitochondrial mutation to pluripotent embryonic stem cells into the mouse to create then a mouse that had that exactly same human mitochondrial variant. And what that does is reduce mitochondrial respiration, oxidative mitochondrial complex 1 by about 60%. That increases decreases the oxidation of NADH, which you can see in the hippocampus. We have high NADH levels, and that increases mitochondrial ROS, and that results in neurological damage. And if we then ask those animals in a behavioral paradigm, here we're looking at the ND6 mutation in which is proline 25 leucine, or another mutation in ND5 complex 1 gene, serine 204 proline that we created. If we look now at the cortical respiration rate, this is control. This is the ND5 mutation and the ND6. And if we now put mice into this stress indicator called a raised zero maze, mice are put here, and then they have to venture out into this more anxiety-producing area. So if we look at the normal mitochondrial DNA, they venture out this level, ND5 this level, and the ND6 this level, exactly proportional to cortical respiration. And again, if we look at their ability to experience depression, which is this forced swim test, we see that the wild type is less prone to depression, ND5 more, ND6 more, and add lithium, wipe out that effect. If we then look at the social interaction, now we're just looking at the ND6. We put them in this field where we have a novel object and a novel mouse. A normal mouse will interact with the novel mouse. A mutant ND6 animal is prone not to, and much more interested in the novel object, and has a hyperaggressive behavior, all associated with changes in the oscillating electromagnetic field that comes out of the brain, which is what's giving those magnetic field oscillations. So we think this is an aspect of the magnetic field oscillation that is called the EEG. So we'd like to finish then by thinking that maybe, in fact, what we're looking at here is a complex array of systems that look at nuclear gene expression through either mutations or oscillation of the epigenome that can affect mitochondrial variants that affect OXPHOS. The environment, whether the calories that we use or how we use those calories, or if we're in fact in outer space and we're in a totally different environment, they're going to modulate the quantum biology of oxidative phosphorylation. It's going to alter increased mitochondrial mutation, cause a progressive decline in mitochondrial DNA function, and that we believe is aging and age-related diseases. And that's going to affect the brain, the heart, the muscle, and the renal system. And that's going to activate the inflammasome because of the release of the DAMPs. And because we're not burning all the carbohydrates and fats, we're going to have what's the equivalent of metabolic syndrome. And the same logic then relates to a whole bunch of different phenomena, cancer, COVID-19, and we believe spaceflight. So I'd like to just finish by, again, my long-term friendship, 3 years friendship with Afshin, my long-term studies with Peter Burke, and more recent interactions with Clarisse, then all of the colleagues in our center that do all of this great work, and our funding sources. And thank you very much for your attention.
Thank you for inviting me again to give a talk, and thanks for. I like going after Doug because everyone becomes a mitochondrial expert after Doug. So now I don't have to really tell you much about mitochondria, but just sort of show the data, and then we can talk about all the nice things Doug has presented. Yeah, and I appreciate the long collaboration and friendship I've had with Doug, because I've learned a lot from him too. I don't think I have to really say why I care about spaceflight, because I think Alistair talked about it, Doug talked about it. And one of the main things is that, as Doug mentioned and Alistair mentioned, that space is definitely an accelerated model for aging and mitochondrial disease, as we're showing. So the space environment, which, again, I think most people here are familiar with, so I'll go through it pretty quickly, is that, you got these 5 hazards that NASA has identified and most people agree with. There's distance, away from Earth, confinement, being in a closed environment can cause all sorts of different stress levels in the body to go up, hostile and closed environment. And the 2 things I really focus on is, the lack of gravity, microgravity, or space radiation, as Doug and Alistair pointed out. And of course, with space radiation, there's 2 types that are dominant. It's the solar particle events, which is occasional. It's not all the time, which is a large acute dose of protons at different energies. And then there's, of course, the galactic cosmic rays, which is the background radiation that we have in space that are just from various events like supernovas and other things that happen that cause these background radiation to happen constantly in space. And as we've alluded to already in the past talk, as Doug talked now, that, all the current experiments that are done is done at the low Earth orbit, which is where the ISS, the previous shuttle experiments were done. And that's, within the Earth's magnetic field of Van Allen. And of course, as we start exploring out to the moon and Mars, you're going to leave this protection of the magnetic field, which reduces the dose of radiation, and you can get the full effect. One thing to this is a kind of a plot here showing the different types of irradiation people are exposed to, either on Earth or in space. The green bars are ones that are exposed to on Earth, and the blue bars are what's been estimated doses we get in space, and it's kind of an estimated dose of a 3-year mission to Mars. But as you might see, you might say, okay, 1 year on Earth's surface, you get this amount of dose, 5.5 millisieverts. But in space shuttle mission, here is about equivalent. But the thing to keep in mind, which I'm going to talk about in the next slides, is that that's not equivalent damage done to the body. Of course, the Fantastic Four had a premonition that back then that being exposed to galactic cosmic rays could cause some defects. They said you're going to turn into superheroes, but I think we have to go back to Marvel and maybe tell them they're wrong. I'm going to tell you why in a second. But if you're not familiar, I'm sure most of you are, there's low LET and there's high LET. Well, LET is the linear energy transfer, meaning the amount of energy an ion will transfer to your cells. And low LET is what we're exposed to on Earth, the gamma, photons and all that stuff. So basically, it's really fast-moving particles. They go through the cell, and the transfer of energy to the cells is not as damaging as these high LET particles, which is now all this stuff that you're exposed to space, from protons to helium to iron particles. Occasionally, you might get some of these high ones, but usually iron particles is the largest you get. So they're slow-moving, or slower moving than the low LET. But what happens is like they deposit all the energy. So it's kind of like a bowling ball. All of a sudden, it sits in the middle of your cell and causes all this energy to be dispersed and cause all this damage to be done. So in reality, what should happen is, in the comic world, this Fantastic Four will be dead right away. So one issue and done. And I guess it's not a very appeasing for comic books and movies. But the other thing that you got to think of is that equivalent dose of like 1 year on Earth to the space shuttle mission that's there, for example, or these other missions. The damage that's estimated is about like 10 times more, or sometimes 5 to 10 times more, depending on what ion you're thinking of, or a combination of ions. So obviously, you might say, okay, you get the same amount of absorbed radiation dose, but this radiation dose is going to cause so much more damage to your mitochondria, for example. So then what's all the impact for health and effects, and risks? And of course, we know there's a bunch of different, there's sex-independent and sex-dependent health risks. And I think Doug made the nice transition talking about reproduction and female reproduction and how might that affect birth, as I'm going to talk about later on in the talk. But, there's obviously things related with, cancer risk, as you mentioned from this space radiation impact, sleep, circadian rhythm issues that Doug had mentioned. And then, of course, there's all these other things. Every part of your body's, from muscle generation, that's more probably microgravity oriented, to bone loss, to, kidney issues, to space-associated neuroocular syndrome, which I'll briefly mention. Doug did a nice job with the eye data to maybe talk about that, which is the issues with vision loss. Anyway, so there's a lot of different health risks. So the type of experiments, again, I think I've presented this previously, but I'm going to really quickly go through the type of experiments that people can do. You could send your mouse or nuts to space. This is the mice up in the ISS. They usually adapt pretty quickly. You don't need to give them some exercise drills because they learn how to really quickly use microgravity as an exercise method for themselves. And then you do simulated experiments on Earth. Typically, we use Brookhaven National Lab, which is a high energy physics collider lab in Long Island, New York. And back in early 2000s, NASA has set up this NASA Space Radiation Laboratory where you can take part of this beam, which is here, and then you could simulate space radiation, iron particles, protons, or a combination of things like galactic cosmic ray, which you have a simulated beam to do. You have these microgravity simulating experiments. Now, you can never truly simulate microgravity on Earth. But, we try to attempt as close as we get. So mice experiments, we do this high limb unloading, which is kind of silly. You suspend the mice by their tail for a few weeks. So you're kind of simulating that unloading of the weight in the back leg. And then fluid shifts, kind of dynamic. So it's not exactly 100%, but it's the best we got. And we could get some kind of relationships to biological changes we get. And then there's also other type of models with collaborators like Seward and Murray are trying to develop, or have developed, I should say. There's other animal models, which I'm not getting into because I'm not using, but people look at Drosophila, C. elegans, they even said zebrafish, they said squid, for all these different types of biology. And we'll look at plants, of course, is a huge area of research that I won't get into, but which we cover a little bit, but and then microbes and other things. So of course, there's this resource that I mentioned is GeneLab, or the Open Science Data Repository now, which has all other things, but GeneLab has all the omics related to spaceflight that people could mine. So that's just a kind of a quick background I mentioned before. and now the spaceflight impact of mitochondria. Doug mentioned this paper. I'm not going to go too much in this paper because I presented in the past, and you could read it. It's a few years old, but these are all the people that were helping, and of course, Doug helped contribute to this paper too. But in a nutshell, I'm just getting the big preview, and Doug had brought it, the main part is that, the group that was here went into GeneLab, mined all the data from different or parts of the mice that were sent. The mice were sent to space, but then the organs that were sequenced from it, they're sacrificed, different cell types, different types of omics from DNA, from RNA to methylation data to protein and metabolites. And lo and behold, when everyone came back and said, what's the top hit of changes that happened, it was the mitochondria and the mitochondrial deficiencies that were occurring there. And then we also had the NASA twin study that I'll show a little bit about what Doug showed that. And then we had all this physiological data. So in a nutshell, basically, this paper showed that mitochondria's function was a pretty good indicator, a central hub of dysregulation that Doug nicely went into. So every 10 years, hence the decadal survey, that's the title, NASA comes out with a decadal survey, what dictates what should happen in the next 10 years of space research within at least the US and within the NASA system. And so this decadal survey was just released last month in September. And one of the key things that came out of it was that people should be focusing on mitochondria, on metabolic energy balance. And here's a little thing highlighted. And of course, they referenced our paper, because I think there's previous papers that talked about mitochondrial dysfunction, but nothing that was unified in a sense. So that was good and made an impact. And some of the areas of researcher focus, which might be pleased, because from which we just learned from Doug and everything he showed, everything that he mentioned is very related to what people are now should be probably one of the key aspects of research, not just speaking, but one of the key aspects of research, which should be outlined according to the decadal survey in the next 10 years. So mitochondrial and things with ROS and ATP and dysfunction having space is a key thing that I think everyone here should be happy to see that would be relevant for everyone's research that we're looking at here. So that's good to see that now people are really paying attention to mitochondria. So now I'm going to show you, most of the data I'm going to show you is going to be unpublished, because I've already shown a lot of the published work, so you can read that. So all the unpublished work, I'm okay to share, and if people want to collaborate, I'm very open with collaborations, and I'm not afraid of people stealing data, because I say, come help us and join us in our venture, these papers, because it'd be fun to have more people involved and more ideas and thoughts. So there's now some data sets available in GeneLab. So this is from Fumi, who's here from Japan. He does a lot of different experiments with JAXA. And this is a data set he actually was able to deposit on the GeneLab source. So now you got one of the, I think, a nice human data set from the cell-free, they call cell-free RNA. So basically, I'll detect the blood, and they looked at the RNA, which includes exosomes and other things, and they made the data available. So this is now running our pipeline that we've developed with Doug, for other work, and now this work. We run a bunch of different analysis, and one thing to look at is now the oxphos. So one thing about this data is the 6 astronauts that were JAXA astronauts, 120 days in the ISS, and this is each astronaut, the group of 6 astronauts are averaged together, which is what you see here for the expression in the heat maps. And you can see this is pre-flight, this is during flight. So if you look at the time, this is like 5 days up to 120 days, which goes 5, 30 days, 60 days, 120 days. And then when they come back from flight, then they got, again, 3 days, 30 dadays, 60 days, 120 days as you go down. So you can see that. And then, this is the full change values compared to flight versus pre-flight, post-flight versus pre-flight, and post-flight versus flight. But the interesting thing, or the one caveat you should keep in mind, these fold changes might not be as accurate at the time, because as you, let's say, collectively get longer in space, your mitochondria is going to get more, as Doug pointed out, it's going to get more and more damage. So you're going to get expression changes happening. So the fold changes might be not as obvious, as things happening as if you look at the individual time points. But that said, and then there's this other data set that with Chris Mason and his postdoc, Jan Kunin, his lab, there's a bunch of things. If you recall, about now almost 2 years ago, Inspiration 4 was the first civilian mission in orbit that did lots of science, and Chris was one of the main PIs involved with that. And there's I think what would we say, about 3,000, 4,000 data points that were generated with all the different omics and physical kinds of the physiological things they did and wearables they had. And so Chris and I actually stay tuned in November, because Chris and I are working with Nature Portfolio Magazine. We're going to put out this huge package of papers, about 50 papers involved, and 25 of them are from Chris and I. There's going to be aspects of mitochondria and other things in there. But, this whole package of papers in Nature is going to show a lot of the data. And then also the nice thing about this all the human data is going to be deposited on GeneLab and be available to the public. For the first time, you're going to get this massive amount of human data from astronaut mission. The one thing to keep in mind, there are 3 days only in space. And I don't know if you can see this plot here, but the ISS is here, which is about 500 and ISS is about 300 something, 60 kilometers above the atmosphere elevation. The ISS, they went up to 390 kilometers, so they were a little, getting closer to the edge of the Van Allen belt. So they were exposed to more radiation, and the equivalent dose they got when you measure from the dosimeters that are there was the 3 days they were at that elevation, they got about 9 months equivalent of radiation to the ISS. So they didn't get as much microgravity effect, but they got, I think one of these guys here went to Chris and said, so are we cooked? And he said, well, at the time, no, but I think their mitochondria might have got cooked a little bit by the radiation. I'll show you why. So interesting thing about JAXA data, if you look at the mitochondrial DNA, as Doug pointed out from the twin study data from the previous paper, one thing that's pretty obvious, you can see at 3 days when they're up there, the elevation is starting to crank up for the mitochondrial DNA in the circulation, but then it gets cranked all the way up from consistently as we go up. Same with the inspiration 4 data. This is the oxphos mitochondrial DNA complex. Now you got the data points we had is you're comparing immediately come from post-flight, which is about three days, about 1 or 2 days to get the samples, and then long-term, which is now 30 days, 40 days, and then like 120 days afterwards. So you can see even, depending on what component of the single cell RNA seq in the blood, depending on what component of the cell, which kind of components, the majority of them, for example, CD4, CD8 cells and other type of T cells had mitochondria enough. So some other cell types didn't have. So the mitochondria DNA complex activity was seen in certain cell types, which is, I think, illuminating. But overall, too, the oxphos suppression that Doug mentioned is pretty universal. For some of the cell types, the long-term aspect would come up. So although when we ask them, like, do you guys feel tired after, fatigue or something that can, be consistent here, they didn't admit it, but you never know. And of course, this is relevant to as Doug showed this nice plot from the twin study data from our previous paper. You can see this is now the previous publication was N equals 1. So now we're actually getting, interesting results from different cohorts of astronauts, different time lengths of that, showing the same exact kind of mitochondrial DNA activity being happening, no matter what setting you look at, the time length, and maybe the dose of radiation, of course, might be a factor there too. Interestingly enough, for the JAXA data post-flight versus the flight data, the oxphos complex starts becoming suppressed a lot more, and maybe long-term damage after we return to Earth. You see this with the inspiration 4 data too. Starts reverting back to a point where you get oxphos suppression happening or inhibition happening, which might be somehow related to what Doug was saying, the overcompensation, and then something happens in damage, and maybe the damage of re-entry and the body reacclimating is now causing this mitochondrial dysregulation to happen. Some other factors, as Doug has shown, the immune factors and HIF, and you can see for the JAXA data, during flight, you can see hypoxia is not as it's a little suppressed, the same as this integrated stress response for the innate immune activity. But after they come back, interestingly enough, even at like 100 on average, and 120 days after spaceflight, you can see hypoxic events are pretty elevated, which is then going back to the oxphos kind of suppression you see here, as you see in the pathway analysis. Same with the integrated stress response, which is related again to what Doug says. So these are interesting factors, and the innate immune response is suppressed, as going back to what Doug was showing too and talking about. So now some ongoing work we're doing too with Doug showed some of this. I'm going to show some of the same plots because Doug's involved with this, but with our good collaborators, Tin, who's a PI at Auburn, or used to be at Reno, but now he's at Auburn University, and his postdocs and graduate students, they developed this nice tool called CPH. It's like a pathway analysis tool. It consists of pathway analysis, which gets rid of some of the bias and things that happen with pathway analysis. But it's a very nice tool. So with them, we've been mining, let's say we used the Mitocarta established pathways that Broad had established for mitochondrial. This is what this is. I'm going to go into the details here. And then we also did the custom pathways that we created with Doug and his postdoc, Joe. So one thing that to keep in mind, I'm going to be a repeat of some of the data that Doug showed, but so I'll go through it quickly, but two caveats to show that, mention that with the data we talking about. Which some of you radiation folks might be familiar with. For example, the strain of mice, The C57 mice tend to be more radioresistant. There's a nice paper here. There's been other papers to show this too. But this paper showed like C57 mice that were BALB/c mice tend to be more radiosensitive. So you got to keep that in mind when the biology changes. And the other interesting thing about these strains of mice, which I'm sure a lot of you are familiar with, is that the mitochondrial DNA versus nuclear DNA ratio in the C57 mice is a lot higher versus the BALB/c mice. So these are the two things, caveats to remember from the mice strains that happen. And a lot of these things that we're going to show, sometimes it's independent of strain, and we showed in our first paper also. And then also consider this is another nice thing from the Japanese Ministry of Environment from the government. They put out, and of course this paper is talking about this, the sensitivity of different organs from the bone marrow to spleen. So there's things where you categorize what Doug was showing. You could think of it this way. So these plots Doug showed, I don't have to really go into them. The only thing I want to add is just to make sure with the eye data. This is the eye data that Doug showed us. He did a very nice explaining, but even despite what Doug was mentioning about all this data, even despite the C57 being more radius resistant than the BALB/c mice, you see the same sort of similar, pretty much similar behavior. Maybe some of the more decreases we see in complex diseases compared to the C57 could be due to the radius sensitivity. Now, what's interesting with the eye data, as Doug had mentioned nicely, is we know that, I'm sure, well, again, to be familiar with this, that there's this phenomenon called space-associated neural uplift syndrome, SANS, for sure. Now, what is conventionally thought is that SANS is probably caused by fluid shifts or physical activity, like the flattening of edema. It could be because of no more gravity, keeping things in check in the body. But I want to propose, or we're going to propose that yeah, sure, you get the physical changes, but most likely maybe all this is driven by mitochondrial activity that we show here, and Doug explained very nicely. So maybe, of course, the physical attribution of the fluid shifts and things could contribute to all this thing with SANS, but could the driver of it truly be mitochondrial dysfunction? And that's what I think we would have to do more experiments and get funding to do more experiments to show that, as Doug was saying. So this is the kidney. I think Doug showed the kidney data. So this is actually interesting. He didn't show the kidney data. So one thing to keep in mind, the kidney data C57 mice that we're looking at, and the interesting about this data set is that there's 2 time points. So you can see the 25 days and the 75 days in the ISS. And with time, with the kidneys to be more of a metabolic rate type organ, you can see that there's that increased shift within the 50 days going with, you see that decrease of complex activity happening. Cell death increases, as Doug nicely explained, the hypoxia goes up, and then you see this immune activity pop up. So with time, you see the impact of it happening in this data set that we're looking at here. And there's this thymus data set Doug showed too. He showed, I think, the top one. This is 2 different experiments that the Japanese Space Agency had done with about the same time point. The only difference was there's just 2 different data sets. So some maybe, of course, experimental variability, even though it might be a case within mouse strains, but overall they show the same pattern. So this is just an explanation, kind of a summary that Joe had provided from Doug's lab very nicely about, what Doug had explained too about the different organs. It's things we haven't showed because it's ongoing work. And so we didn't show the muscle data, but overall in our first paper, we showed a lot of the mitochondrial activity was completely suppressed in the muscle, which makes sense with the muscle degeneration. And we see that here, of course, with the data we're analyzing. And this is just kind of the summary of it, as Doug mentioned. So this is going that, but then the idea is sure, we got all this damage caused by can we mitigate it? There's some ideas, as Doug had mentioned, with mitochondrial supplements we could have, and we didn't mention as much. Something I'm just going to really quickly go over because I presented this at our last time in February, I think, or was it February when I was here? But the microRNA and countermeasures. So microRNAs, as everyone knows, is single, a small non-coding RNAs. They don't translate to proteins. They're very small. A single microRNA can regulate hundreds to thousands of messenger RNA or genes, and due to their stability, their size, they can float in your body either freely, meaning with a protein attached to them, or they can be packed in exosomes or in lipids. And they're known to be involved in good things, all healthy aspects of life, as any kind of genes and proteins are, and then also all aspects of the disease. And the beautiful thing about microRNAs is that, just like everything else, they're found in all organisms, but they're highly conserved in many organisms, especially between mice and humans, which maybe in the microRNA world, they're saying that humans are not too different from mice, whether you like it or not. And the interesting thing is that microRNAs can obviously directly regulate mitochondrial, but binding to mitochondrial-related genes, or mitochondria themselves actually produce their own microRNAs called mitomeres. So they're basically microRNAs that translocate from the nucleus to the mitochondria, and they're and mitochondria has actually microRNAs that are essential for their efficiency and their function. But then, of course, when damage is caused to the mitochondria, there's mitomeres that are produced that are will cause that detrimental damage and loop happening. So in our initial one of our papers here, we published in that cell package that maybe somebody's familiar with. We identified circulating microRNA signature and confirmed this. And this is the first experiment by inhibiting these a set of 3 microRNAs that we said are associated with cardiovascular events. Can actually mitigate the radiation damage completely. And this is an experiment. We go to Brookhaven National Lab, irradiate this 3D microvascular cell structure, and then we applied the integumenters as a pre-treatment, as a countermeasure before irradiation. And these 3 microRNAs, which I'm not going to show most of the data because I've shown it previously to people, is that they're heavily involved with mitochondrial function too. And the 3 microRNAs is this miR16, miR-LET7A, and miR-125B. You can look that up. And this is now a paper that's part of that big Nature package that Chris Mason and I are putting together. We're sending back the revision soon, but basically in a nutshell, we actually went in deep into the biology and saw indeed that when you inhibit these microRNAs, you get this rescue that happens of the integument is very reproducible. And then also the functions, like for example, when you use this and it completely rescues it, it actually rescues the inflammatory and immune functions that are the radiation is causing, and also rescues some mitochondrial functions we show in the paper. So short answer, yes, but and then again, the decadal surveys talking about the importance of looking at things like exosomes and microRNAs is probably an area of research where people should focus on. So just refer to our paper here again, which is great to see. So I guess next 10 years, whether you're looking at exosomes or even cell-free like mitochondrial DNA, they're like, this might be an important way for countermeasures people should explore. So that's another kind of plug to say, let's in the next 10 years push forward with some of this type of research. One thing to keep in mind is that that doing this kind of treatment, of course, need more funding, but also the impact of it might be a little further out because to do these integument treatments, we need to go through the proper, clinical trials, preclinical trials, which takes time, which we're trying to do. But obviously, this is not going to happen next year. This probably in the next 5-10 years, potentially, this could be a countermeasure. We could maybe improve mitochondrial functions for once we show it's maybe effective for humans. Now, something that Doug alluded to before the last question, and the last question is reproductive system. Now, our specific mitochondrial impact in space in females, which is nice. I don't think he was aware I was going to talk about this. So it was nice that he mentioned. And of course, we know Doug's seminal work that back with this paper that he actually shown that mitochondrial DNA inheritance is from the maternal side, which I guess, as Doug puts it too, nature decided men are sort of useless and the women are the key to provide the bioenergetics for our next generation of people and kids. So that's a way of nature saying women are stronger than men, I think. But keep that in mind, as Doug mentioned. And also, as Doug mentioned too, as I keep this in mind that, that we don't understand much of what's happening with the female reproduction system, what's happening with the, mitochondrial changes that might happen that might impact birth defects or female, mitochondrial DNA that could be passed and cause mutations, and maybe that lineage could be passed on to future generations and cause a detrimental effect that we don't want. Again, something I bring up again, the Kale survey they actually talk about, because one of the things to keep in mind in the past decades of research, there hasn't been much, unfortunately, research done with there has been some good papers that have come out, but they're not much. There should be more to it. And so they're talking about here, and so that of course we should pay attention to reproduction and female biology. It should be one of the key now that there's actually more than 50% of the people going to space are females, which is great to see. But what does that mean for, let's say, if a woman goes to space, comes back and wants to give birth, is the long-lasting damage caused, say, to your mitochondrial DNA, is that going to be potentially causing birth defects in the future? So the short answer is probably. So that's not too good to hear, but so working with some other free databases, so like free, there's this NIAID, the National Institute of Aging and Infectious Disease from NIH. They have this nice resource called MPORT. And MPORT has all this other data related to immunology, aging, and all this stuff. So, this is so to be creative is, can we look at data sets which are associated with, birth defects or kind of female related issues in birth, and they're related back to data sets we have from, from space related female activity that we know. whether it's mice related or humans. So luckily on the import data set, there's this one data set on that they looked at microRNAs. This microRNAs I like. There isn't much there is mitochondrial stuff, but we have to look at that later too. But this data set was really nice because they were looking at maternal plasma from microRNAs from maternal plasma that is associated with this thing called disease small for gestational age fetuses, meaning basically women who give birth to kids who are actually smaller than they should be, and could cause birth defects or different kind of detrimental things to the body, even a kind of mitochondrial bioenergetic issues that if you read papers about that. So this is kind of an issue that is concerning when that happens. So in this work that there's papers here that are looking at different microRNAs related to this and thinking, is there a circulating factor? Just like I propose there probably are a lot of circulating factors you can think of. So then are these microRNAs that are associated with SGA and are elevated in the plasma, but are these things elevated in the plasma during spaceflight? And then when they return to Earth, can this potentially be an issue of birth defects and mitochondrial related damage? So going to the free resource, the Gene Lab or Open Science Data repository, there of course is a microRNA thing. This is actually an experiment I've done, so I knew I know going in there I was biased. I know there's an experiment with microRNAs with plasma. This is a simulated experiment that mice hind limb set up and then radiated at Brookhaven, and 24 hours later we sacrificed the mice with these different kind of radiation conditions. And just to orientate you is that this 0.5 great galactic cosmic ray simulated beam this is the ion structure, and this is sort of the estimate is, let's say if someone goes to Mars and comes back. Now we don't have any magnetic field here, so unfortunately that was not part of the experiment and hard to do with these kind of experiments at Brookhaven. But we also had like a simulated solar particle event, which would be 1 gray at these proton different energies. And then there's this other experiment here with microRNAs on the peripheral blood lymphocytes. It's not exactly 1-to-1 for plasma, but I included it anyway just to see what happens. So in a long story short, is that from the SGA, from the data set from the woman who giving the birth defects here, I looked at the microRNA profile and said, what microRNAs are related to these data sets here? What crosses over? So we get a handful of microRNAs. This is a heat map showing that. And of course, you're going to find some microRNAs that go in opposite regulation. But what was really nice to see is that a set of microRNAs here in the green bar circle, and this is a correlation plot for these microRNAs, are highly correlated between the women that are having this SGA, birth defect, and the mice that were exposed to these different conditions, or their hind limb setup, just microgravity event, or the galactic cosmic ray, with or without the hind limb setup. Now you're going to say, well, these are mouse, these are humans. Does it really translate? Well, this is, and some of these microRNAs are already identified for my microsignature, and I've shown that it doesn't indeed correlate with humans in space. But, indeed, with my good collaborator and friend Paco, who's at the University of Lisbon, he performed a homology and to look at basically the similarity or conservation between these microRNAs between mice and humans. And indeed, the mature microRNAs are 100% conserved between all these ones that are identified. Interestingly enough, the precursor micros, if you're familiar with microRNA world, is that you got the precursor micros, which is this longer strand of RNA, the hairpin loop. And then once the mature microRNA happens through a big process where the microRNAs are clipped, then it becomes the mature process. They could become this 22 nucleotide region. So the precursor ones, they don't necessarily see as much change, but it's harder to be conserved. But amazingly enough, the conservation for the majority of microRNAs are pretty close, either 100% or above, 88 to 90%. So indeed, these microRNAs, that's a good thing I mentioned. Remember, microRNAs are very highly conserved between mice and humans. So that means what you do in mice translates well to humans, at least in the microRNA world. So what are the health risks associated with these microRNAs that are common between the birth defects? So of course, pregnancy is up there, we see. Cancer, there's a bunch of other things. But what's interesting now are the pathways. So of course, mitochondria pops up. I shouldn't say of course, I was happy to see that mitochondria popped up because I didn't know if these microRNAs would be affecting mitochondria. But indeed, these microRNAs are actually involved with mitochondrial dysfunction, glucose metabolism, which goes back to what Doug was saying, that oxphos balance, glucose going up in space, carbohydrate and autophagy. And there's other, of course, factors like aging you might see. Of course, aging, it might be a issue in space. Accelerated aging, as Doug pointed out, other things like DNA damage and other factors. What's interesting is that now if you do a kind of a global analysis of the mitochondrial, this is looking at mitopathway, and I'm going to do a custom mitochondrial pathway too, is that, in SGA, these are all significantly regulated pathways. So, but you see this x-axis here is showing whatever's positive here predicts this pathways for the microRNAs are being activated. If it was going up, so it'd be inhibited. But what's interesting is that with the SGA, oh, I forgot to mention this time with the cohort here with the SGAs, they had 3 different time points from gestational period. I did analysis to group them together at time independent, time dependent, and you can see there is some kind of time dependent factors happening at the mitochondria. But what's interesting is that with SGA alone, you get significant, but slight increases from the microRNAs causing like oxphos dysregulation happening metabolism. Now in space simulated experiments, we get this huge dysregulation happening because the circulating factors is happening in the plasma. So if this is happening, and this is all you need to cause SGA, imagine if, let's say, people come back, and we've shown that, or particularly females come back from space, and we've shown that some of this, the microRNAs do not go back to normal levels from our previous work. And these micros are still circulating in the blood, causing this mitochondrial dysregulation to happen. This is associated with SGA ones. That would be kind of concerning if you, let's say, planning to get birth after you've been in space for a period of time, and these things are, be associated with space-related birth defects. So then the idea is, can we potentially have countermeasures again to, block this? So for this type of data, again, this is really preliminary work. This is working on the manuscript, so we haven't done actual experiments to block these microRNAs and see what happens in space. But with my good friend and collaborator Diego Galeano, who's at the University of Centurion in Paraguay, this manuscript's currently under review. But we developed this kind of deep learning, machine learning framework to predict basically what small molecule drugs, FDA-approved small molecule drugs, can target microRNA. So in a nutshell, what he's very clever at doing this. He mined the small molecule drug banks, and then looked at the chemical similarity between that and the microRNA sequences. And within that, basically, it's kind of like if you watching Netflix, and Netflix wants to predict eventually what shows that you like to watch. And what it does is it gathers, your similarity of viewing structure over time to millions of other people's viewing habits. And that's a machine learning tool they use, basically are able to predict from your behavior to the millions out there how things can be related, and what, let's say, if you like comedies versus sci-fi or vice versa. And this is the same kind of idea here, looking at that. So long story short here is that some of the microRNAs, this is just showing from this microRNA tissue atlas database, that what kind of the basal level you might find in humans in these microRNAs are supposedly healthy, in quotes. That's what they're looking at, healthy tissues. But what's interesting now, when you predict from his FDA-approved drugs, this is the kind of the drug indication, meaning what kind of behavior the drug's having, some of these related with female-related issues, and then the mode of action of the drugs, so like estrogen, glucocorticoids, which is related to mitochondrial activity, and vitamin D, which we know is heavily involved with mitochondria, and so on. And then if we focus in on the specific drugs, so of course, there's a bunch of drugs that he had predicted related, but 2 drugs are commonly shown to be targeting all 13 of these, or in this case, there's 10 that showed up microRNAs within the databases available and the resources. Interesting enough, this one microRNA, this one drug, FDA-approved drug, these are both actually, some of these are different clinical phases, but these 2 actually launched and FDA-approved already. So this one is triamcinolone, which is a glucocorticoid, which hasn't been used previously for like birth defects, but it's more like conditions like eczema and cirrhosis and other kinds of skin-related issues and other factors, which could be a potential novel thing that people could potentially use, maybe target these microRNAs and prevent maybe this SGA-related space issue. But interesting enough, this parafluorocarbon, parafluorodecaline, has been used for a while. It's kind of an artificial blood product that's been very effective. And as you can imagine, this being what it does, it can actually improve your mitochondrial functions, which is, because it provides extra oxygen and provide healing. What's interesting when you start looking at the literature related birth defects, people actually have already considered using this drug as a potential way to mitigate some of this issues with preterm infants, and then some gas exchanges that have been mitochondrial respiratory type related diseases. These experiments done with lambs. And what's interesting, as you start looking at literature related with how they're related to mitochondria, again, as you might expect, the parafluorocarbons, emulsion is a good way to provide oxygen and allow for efficiency and improved mitochondrial oxyphosphorus, as paper shows and other papers have. So this could be a potential way to maybe mitigate the birth defects and rescue the mitochondrial damage done by space, and at least for the female reproductive angle. And that, in a nutshell, is I could talk more, but I won't. But this is I think hopefully Doug did a really good job of going into the details of mitochondria, but going into a lot of the data associated with it. And then, of course, I put in the whole microRNA angle that potentially could be a very good biomarker and way and a countermeasure to rescue the mitochondrial defects of damage that is done. So thank you very much for having me present this.